Primary research article we reviewed was SIRT-XRN2 suppresses tumor progression driven by histone lactylation through activating the Hippo pathway in human bladder cancer. Before discussing the research paper, it's important to know some background information about bladder cancer, circRNAs, the Hippo pathway, and histone lactylation. Bladder cancer is the fourth most common malignant tumor. It is also the 13th leading cause of death in cancer patients worldwide. Because of the prevalence and severity of bladder cancer, it's important to conduct more in-depth investigations into the tumorigenesis and progression of bladder cancer. circRNAs are long non-coding RNAs. They have a long covalent loop structure that makes them more stable than linear RNAs. Previous studies have shown that circRNAs regulate the occurrence and development of cancer. For example, some circRNAs regulate bladder cancer progression by acting as miRNA sponges. It has also been observed that circXRN2, which is a specific type of circRNA, is downregulated in bladder cancer tissues and cell lines. The Hippo pathway regulates numerous biological processes, including organ development, the regeneration of tissue, cell proliferation, and immune regulation. The Hippo signaling pathway consists of a kinase cascade and two executors called TAZ and YAP. A protein called LATS1 activates the signaling axis of the Hippo pathway by phosphorylating the two executors TAZ and YAP. It has been demonstrated that the aberrant status of the Hippo pathway in human cancer promotes tumorigenesis and metastatic progression. Histone lactylation regulates cell biological functions by activating the downstream transcription and expression regions of a gene. Previous research has discovered that histone lactylation drives tumor progression in human cancer. Biological question. Current studies rarely investigate how the novel molecular mechanisms and regulatory models of circRNAs affect human bladder cancer development and progression, which has created an urgent need for further studies. The biological question addressed in this research article was, can a novel molecular mechanism of circRNAs suppress the progression of bladder cancer? On the biological question, the researchers hypothesized that circXRN2 might inhibit proliferation of tumor cells, that circXRN2 might modulate LATS1 protein levels via post-translational modification, that SPOP might participate in the degradation of LTS1 protein regulated by circXRN2, and that circXRN2 and SPOP might competitively interact with LTS1 protein to regulate its degradation.
Here is the assays and methods portion of our research paper. First, we'll talk about the RNA immunoprecipitation assay, or the RIP assay. The RIP assay uses antibodies to help isolate and extract RNA. Here, pretreated magnetic protein A and G beads were exposed to specific antibodies, which were then collected to use in subsequent assays. Next, we'll talk about the cleavage under targets and tagmentation assay, or the cut and tag assay. The cut and tag assay is used when the sample size is very small. Here, H3K181A antibody and a secondary antibody, as well as hyperactive PATN5 transposase, were incubated in cells where hyperactive PATN5 precisely cut off DNA fragments with the target protein. The DNA fragments were ligated and amplified with PCR to then sequence. Next, the Agilent Seahorse XFE24, or Seahorse Metabolic Analyzer Glycolytic Rate Assay, is useful because it is non-invasive and used for real-time glycolysis analysis in living cells using calculations to determine the glycolytic proton efflux rate, basal glycolysis rate, and the compensatory glycolysis rate. This research used Agilent Seahorse XFE24 to determine the glycolytic rate for multiple cells in the study. The protein immunoprecipitation assay, or IP assay, is used to remove proteins bound to antibodies as well as to measure outside interactions with the specific protein. This process takes advantage of the specific affinity between antibody and protein. Here, the precipitate was collected from the supernatant fluid after being incubated with specific proteins. The cell counting kit 8, or CCK8 assay. The CCK8 assay is useful in determining cell viability. In this research, cells were seeded into 96 well plates and 10 microliters of CCK8 added. After incubation, absorbance was measured to be 450 nanometers and cell viability was calculated. A colony formation assay is used to determine whether a cell can reproduce and form a colony. In this research, 2,000 individual cells were cultured in 12-well plates, and colonies formed after 1 week were fixed, stained, and counted. The subcutaneous tumorigenesis model is used to implant tumor cells under the skin of mice. The resulting tumors are measured over a period of time. Here, tumor cells were implanted into 4-week-old nude mice, and tumor growth was measured for 7 days. The tumors were harvested after 35 days, and their mass was recorded. The tail vein lung metastasis model consists of intravenous injection of tumor cells with an affinity for lung tissue via the tail vein of a mouse. Here, T24 cells, or carcinoma of the bladder, were injected into 6-week-old nude mice. Lung tissues were sampled 50 days later, fixed, and sent to be stained with hematoxylin and eosin.
This is Andrew, and I will be doing the results. LATS1 is downregulated in bladder cancer and interacts with SIRC-XRN2. It is regulated in bladder cancer tissues compared to normal. It verified the expression level of SIRC-XRN2 and LATS1 binding affinity. These results suggest that SIRC-XRN2 could be involved in the modulation of the Hippo signaling pathway. SIRC-XRN2 inhibits the proliferation and migration of bladder cancer cells in vitro and in vivo. Sanger sequencing confirms the conjunctive site sequence of SIRC-XRN2. SIRC-XRN2 could only be amplified by divergent primers in cDNA. This was confirmed by the loop structure. SIRC-XRN2 is mainly localized in the cytoplasm, and it can demonstrate inhibitory effects. It can also reduce the growth rate and weight of tumors. These results suggest that SIRC-XRN2 suppresses the proliferation and migration of bladder cancer cells. SIRC-XRN2 activates the Hippo pathway by stabilizing LATS1 to regulate bladder cancer progression and glycolysis and lactate production. Protein levels of the LATS1, TAZ, and YAP, and the intercellular location of TAZ/YAP was confirmed by immunofluorescence. Depletion of LATS1 reversed the inhibitory effects on cell viability and proliferation induced by SIRC-XRN2. The SIRC-XRN2 LATS-S1 axis inhibits tumor growth and metastasis of bladder cancer cells in vivo. Overexpression of SIRC-XRN2 significantly reduced the tumor growth rate, and knockdown of LATS1 significantly attenuated the inhibitory effect caused by SIRC-XRN2. Overexpression of SIRC-XRN2 significantly reduced lung metastatic nodules compared with the normal control group, while tumor nodules were significantly increased in cells treated with SIRC-XRN2 overexpression, along with the knockdown of LATS1. To conclude, SIRC-XRN2 suppresses tumor progression mediated by H3K18 lactylation in human bladder cancer by activating the Hippo signaling pathway.
This is the interpretation of the experimental results. From the experiments on circRNA, it is very clear that they serve an important role in regulating cell activities despite being non-coding RNA. In the bladder cancer cells, the researchers found dysfunctional SIRC-RNX2, which suggests that it is involved in suppressing cell proliferation and preventing tumor progression. SIRC-RNX2 seems to do this by preventing LATS1 from being ubiquitinated and degraded by binding to LATS1 degrons, thus increasing LATS1 concentrations in the cell. LATS1 is an essential factor in the Hippo signaling cascade. SIRC-RNX2's modulation of LATS1 seems to heavily influence the Hippo pathway. In the experiments, SIRC-RNX2 was dysfunctional. The Hippo pathway doesn't activate properly, and cancer cells spread rapidly. But when SIRC-RNX2 was reactivated in certain cells, the cancer cells had significantly less proliferation and migration. This suggests that SIRC-RNX2 is a driver of the Hippo cascade pathway. The researchers found that disruption of this pathway caused metabolic abnormalities in the cell, which may be contributing to the increased proliferation rates of cancer cells. SERC-RNX2 also inhibits H3K18 lactylation through the Hippo pathway. H3K18 is a known oncogene in bladder cancer. The researchers did a great job overall drawing these interpretations from the experiments, and their data seems to back up their conclusions well.
Conclusion. The researchers concluded that SERC-XRN2 suppresses tumor progression mediated by H3K18 lactylation in human bladder cancer by activating the Hippo signaling pathway. We agree with the researchers' conclusion because the results of the study's experiments were consistent with trends in previous experiments, their predictions for the experiments were reasonable and matched the results, the process of identifying SERC-XRN2's role in bladder cancer tumor formation and progression was gradual and thorough, and the in vivo and in vitro tests for determining if SERC-XRN2 LATS1 inhibited tumor growth and metastasis of bladder cancer cells had similar results. In addition to this, the researchers used good experimental practices, including having well-formulated hypotheses, using control and experimental groups, and testing in vivo and in vitro. Future ideas. SERC-RNAs such as SERC-XRN2 can be studied further to find precise ways to influence the Hippo pathway and provide an effective treatment for bladder cancer. This research can then be used to find SERC-RNAs that affect regulatory pathways and suppress tumor progression and metastasis for other types of cancer.